Okay, dear students, we are going to talk about complement system. Now, what is complement system? Why it is called complement system? Because it complement or enhance the activity of antibodies. है ना? And antibodies are produced against any invading organisms, or specific antibodies are produced against specific antigen of any bacteria or viruses or any exogenous substances. They can directly attack the invading agents, or they attack through complement system. Now let's say, suppose this is bacteria, this is your bacteria. Now bacteria has antigen present on the cell membrane, and our immune system produces antibody against that specific antigen. These are the specific antibodies. They attack on the specific antigen of bacteria. Now as soon as they bind to the specific antigen, and this portion is called FAB, antigen binding site, and this portion is called FC segment. As soon as antibody bind to the specific antigen present on the cell membrane of bacteria, this FC segment gets activated. Now when this FC segment gets activated, certain some neutrophils and some monocytes, they have receptor for this FC segment. So this FC segment bind to this receptor or this receptor of neutrophils, and that neutrophils and monocyte or macrophages, they effectively phagocytose bacteria. This process is called opsonin or opsonization. What is opsonin? The molecules which enhance the phagocytosis activity, that is called opsonin molecules. This process is called opsonization. Okay, so antibody, either they direct attack on invading agents through agglutination, or precipitation, or they neutralization and cytolysis. This is the direct attack on invading organism. But this antibody can also destroy invading organism, bacteria or viruses or any exogenous substances through indirect attack through complement system. Now let's say, what are complement proteins? Most of the proteins are produced by which organ? Liver. Now liver constantly produce complement proteins in our circulation, and complement proteins C1 to C9 and B and D. Total 11 complement proteins are to be identified by scientists. So these are the complement proteins which are produced constantly by liver. They are already present in our circulation, but suppose here there are our normal tissue, and this tissue are invaded by, suppose this is your bacteria, and inflammatory process occur, vasodilation, interendothelial gaps are formed, and these complement proteins leak into at the site of infection, complement proteins. Now let's say, suppose this is your bacteria. This bacteria has specific antigen present on their cell wall, and B cell, with the help of T helper 2 cells, they convert the B cell into plasma cells, and that plasma cells produces specific antibody against these specific antigens. है This FAB segment of the antibody bind to these antigen. Now as soon as the FAB segment they bind to the antigens, FC segments get activated, and when as soon as the FC segment gets activated, complement protein number 1, which attach and activated to the FC segment, C1 complement protein, which also leak through the interendothelial gaps. Now this C1 protein also release some enzymes which activate another complement protein, which is called C4. Now this C4 complement protein has two components, A component and B component. A component gets released and B component remain bound to the C1. So C1 component activates C4 complement protein, and C4 complement protein has two component, A and B component. A component will be released into extracellular matrix and B component remain bound to this C1 complement protein. Now this C1 and C4b together it is called C2 convertase. C1 and C4 together they are called C2 convertase. Why? Because they now activate C2 complement protein. Now again this C2 complement protein has two component, A and B component. A component will be released, go away, and B component remain attached or remain bound to the C2 convertase complex. Now these three together, C1, C4b and C2b, they together are called C3 convertase. And they activate C3 complement protein. Again A component will move away and B component remain attached to the C3 convertase complex. Now this C1, 4b, 2b and 3b, these components again called C5 convertase. Why? Because it is going to activate C5 complement protein. C5 A component will be released and C5b remain bound. Now this C5b activates C6, C7, C8, C9 complement protein. So this 5, 6, 7, 8 and 9, they are together called membrane attack complex, which attack cell membrane of bacteria. These 5 proteins make up pentameric proteins. Okay, let's say suppose this is your protein complex formed by the 5, 6, 7, 8 and 9. 5, 6, 7, 8 and 9. This pentameric protein has a central pore. This pentameric protein gets implanted into the cell membrane. Now, as soon as this pentameric protein gets implanted on the cell membrane, all the materials of the bacteria will leak out. And bacteria got osmotic shock when all the materials, all the major cations, proteins, all will be leak out through this channel, membrane attack complex, and this bacteria got osmotic shock and bacteria dies. So this is the classical pathway of complement system. Okay, now look, let's revise. These are the what antigen present on the cell membrane of bacteria. Our immune system produces antibody against this antigen. Fc segment gets activated, which activates C1 complement protein. C1 complement protein activates C4b proteins, complement proteins. C4b activates C2b. This C1, 4b and 2b, they activate C3b. C3b activate C5b, and 5b finally activates C6, 7, 8 and 9, which is called membrane attack complex. This is classical pathway of the complement system. It is the indirect killing by the antibody through complement system, classical pathway. Now what is what is alternative pathway? Which pathway? Alternative pathway. Now certain bacteria, certain gram negative bacteria, there are two types of bacteria, gram negative bacteria and gram positive bacteria. They are also called bacilli. Now these bacteria has specific proteins present on their cell membrane, and these proteins are called properdin. They are made up of lipopolysaccharide. These are endotoxins. Now another protein, complementary protein, which is already present in our body, when it leak out at the site of infection, that protein, which is C3b, which bind to this properdin, it stimulate directly without antibody, which complement protein, it activate C3b. Now C3b activate another complement protein which is called B, capital B. As soon as this capital B gets activated, A component will be released, B component remain bound. This C3b and capital BB again activate C3b protein. C3A component will move away, remain detached, and C3b remain bound to this complex. And rest of the story will be the same. C3b activates C5, A component will move away, and which activates C6, C7, C8 and C9. And this is called membrane attack complex, and which makes whole. And this pentameric protein, it makes channel and that gets implanted in the cell membrane. And these proteins are also called perforin. It perforate cell membrane. That's why it is called perforin. All the substance of the bacteria will leak out and bacteria will die. Okay, this is alternative pathway. Okay, what is difference in classical pathway and alternative pathway? In classical pathway, antibody Fc segment gets activated, which in turn activate the series of complement proteins. But here protein properdin, which is lipopolysaccharide, which is present on the gram-negative bacteria. So this is also called properdin pathway, alternative pathway or properdin pathway. So these are the two pathways. First is classical pathway, second is alternative pathway or properdin pathway. Now third pathway is mannose binding lectin pathway, MBL pathway. Mannose binding lectin pathway, MBL. Let's say suppose this is bacteria. Some bacteria has mannose protein present on cell membrane. This cell membrane of gram-negative bacteria, they have some specific protein which is called mannose binding lectin. Now to which complement number 3 protein, which gets attached and gets activated, and it activate series of protein. Which protein? C5, C6, C7, C8, C9 and bang. Membrane attack complex. This is third pathway, which is called MBL pathway or mannose binding protein. Okay, so basically there are three pathways. First pathway is classical pathway. Okay, what happens in classical pathway? Antibody binds to antigens, Fc segment gets activated, which activates C1, C1 in turn activates C4, C4 activates C2, A component will be move away and B remain bound. 1, 4 and 2 activate C3B, A will move away. C3 in turn activates C5, C6, C7, C8 and C9, which attack your bacterial cell wall. This is classical pathway. What happen in alternative pathway? Properdin protein which are present on the cell wall, through which directly C3B gets activated. C3B activate capital B, A component will move away, BB component remain bound to C3B, again it activates C3B and rest of the story is same. C5, C6, C7, C8, C9 and perforin. This pentameric protein which are also called perforin. They are pentameric protein which gets implanted in the cell wall, make a hole, all the substance of the bacteria will leak out and bacteria will die. Structural integrity of bacteria will be lost. So these are the three pathways of the complement system. Okay, third pathway, MBL pathway, where mannose binding lectin pathway, and which trigger the series of complement proteins C3b, C5b, C6, C7, C8 and C9. Okay, first pathway is classical pathway, second is alternative, and third pathway is MBL, mannose binding lectin pathway. Okay. Now, the A component which are released during the process of activation of this complement system, they act as C3A or C4A, C2A, C5A. All these proteins act as a chemoattractant. Chemoattractive to neutrophils and monocyte. Okay, that's how antibody indirectly kill through complement system, direct activation of the complement system through the alternative pathway, and third is the MBL pathway, which is mannose binding lectin pathway. Because this is all about complement system, this is the most important topic of the immunity. So let's check. This is given in Hindu Purana. Attack on antigen through complement system, classical pathway, and this is your what? This is flow chart given, classical as well as alternative pathway. Classical pathway through opsonization, lysis, agglutination, chemotaxis, neutralization, and it also activate mast cell and basophils. And alternative pathway, which is also called properdine pathway, and third pathway is mannose binding lectin pathway. Okay, so this is all about complement system.